Hello, welcome back. This is the Division of Education, Innovation and Energy e-learning program. And I am Ms. Jamilla Bacchus, your facilitator for CAPE Biology. In particular today, we will be looking at Unit 2 Biology, and we will be reviewing one of the topics you would have done in Module 1, which is respiration. So now that we are finished with glycolysis, the next stage is what we call the link reaction. So once glycolysis has taken place in the cytoplasm, only if oxygen is present, this next stage will take place. And this is where the product of glycolysis, which is pyruvate, will now move into the mitochondria. If there is no oxygen present, then you will not have the mitochondria coming into play. So what we call anaerobes, organisms that live in swamps and so on, that have very little oxygen present and so on, they can switch to, some of them totally carry out anaerobic respiration. So the mitochondria will not come into place. So within this link reaction, let's do a little drawing here. So you have pyruvate here. And pyruvate is a 3-carbon molecule. And you have to go from pyruvate to another molecule, which we call acetyl. And acetyl is a 2-carbon molecule. It has 2 carbons in its structure. So to go from this 3-carbon to 2-carbon molecules, what we can do will be to remove one of the carbons in the form of carbon dioxide. So when we have carbon dioxide being removed, because we are removing one of the carbons, so carbon dioxide is coming off because we're taking it from this 3-carbon molecule. So here we are removing carbon dioxide, and so we go to a 2-carbon molecule. But what we would also have, as we have removed one of the carbon, you will also find that there are hydrogens that will now be able to reduce NAD to become NADH. So you will have NAD becoming NADH plus H plus, right? So what is happening here? It's reduction that is happening. The NAD is being reduced to NADH. This will have significance. Remember to put in your arrowhead, very, very important within these reactions that you put in your arrowhead. This product, NADH, will now go into the electron transport chain, where, as we said, for every 1 NADH, you're going to yield 3 moles of ATP. And because we would have had 2 pyruvate molecules, then we're going to have 2 NADH being produced. And so, in truth and in fact, we will get 6 molecules from ATP just from this stage here. Now, the next stage will be for the acetyl to meet with the molecule that would be in the Krebs cycle. So from here, so this here is called the link reaction, L-I-N-K reaction. And this link reaction is simply bridging glycolysis with the Krebs cycle. Remember, this is happening within the mitochondria. This is not happening in the cytoplasm. Once pyruvate has been formed and oxygen is present within the cell, this link reaction, the pyruvate will go into the mitochondria, and this link reaction will occur in the matrix of the mitochondria. And as we see here, so acetyl will now be added to coenzyme A. Coenzyme A, it's important for coenzyme A to be able to carry the acetyl, this 2-carbon molecule, into this Krebs cycle, also known as the tricarboxylic acid cycle or the citric acid cycle. You see here oxaloacetate. You are not required to know the names of these molecules. You are required to simply have an appreciation of what is happening at different points. At CAPE level, you are not asked to name these molecules. That is very important. At university level, you do. Right? So this is a 4-carbon molecule. And when this is added to this acetyl coenzyme A, so acetyl is the 2-carbon molecule, and coenzyme A is helping it to make the link. So this molecule comes around, and this one comes in, and so they now link. And you now have this molecule, which is called citrate, which is a 6-carbon molecule. So the 4-carbon from here and the 2-carbon from here, which came from glycolysis, would have now formed the 6-carbon molecule. The coenzyme A is no longer needed, and so this simply exits, as you can see here. So you simply have this 6-carbon molecule, and from the 6-carbon molecule, you are going to have the loss of 1 carbon dioxide, but remember, everything we are multiplying by 2 because we would have had 2, remember from glycolysis, 2 glyceraldehyde 3-phosphate, 2 pyruvate, so we now have 2 carbon dioxide, 1 from each of the molecules being removed. You also have, so here now you have a 5-carbon molecule, right? And eventually you have another carbon dioxide being lost, 1 for each molecule. You also will have, hold on a sec. Right, so you have a 1 carbon dioxide for each molecule being lost, and so you will end up with a 4-carbon molecule. Now what is happening along the way? You're going to have some hydrogens being made available, some hydrogen and some energy being made available to reduce NAD. So again, we have in the reduction of NAD to NADH. Remember, you cannot get these energy at this point. They will be eventually released when we go to the electron transport chain. And so this will be the product. Again, you see it here. So you're seeing 1 per molecule, 2 per molecule, and you see the last one here. So again, you have to take note of that. You're required to know how many reduced NAD will be in the Krebs cycle. And you're seeing 1 FAD form for every molecule. So for every molecule, you have 3 NADH being formed, 3 reduced NAD, and 1 reduced FAD. Again, these are going to go to the electron transfer chain. Since you had 2 molecules, you will then have 6 NADH and 2 FADH. Good? And so we can multiply them according to how much ATP they will yield, and that is how we calculate how much energy will be available. There is also 1 direct production of ATP, as you're seeing here. One of the reactions will yield the direct production of ATP. Again, going back to your definition of substrate level phosphorylation, this is what is happening here. A high yielding substrate with phosphate is going to now allow ADP to link to the phosphate to form ATP. And so for every acetyl molecule that you started off with, you're going to get 1 ATP, so you have 2 ATP being formed because you had 2 molecules. Keep reminding you of that, right? So this is the Krebs cycle. Again, you don't have to know the exact name of all of these molecules, but you should have a general appreciation of what is happening, where it's happening, where carbon dioxide is being lost, where NAD is being reduced, where FAD is being reduced, and where you have the direct formation of ATP. This now is the electron transport chain. And here we are seeing representation of the membranes. So you see in this, we know membrane structure. You have the phospholipids, the bilayer, and you're seeing these molecules between the bilayer. They are proteins, and they can accept electrons, and they can also facilitate the transport of hydrogen ions across, both of which happens when the electron acceptors that we met earlier, the coenzymes would have picked up the electrons and the energy in the earlier stages. And now this is where they're going to come to finally release the energy to form ATP. So here you are seeing it again. What I want you to note is this structure here. This structure here is ATPase. It is a protein that is embedded in the membrane, and this is responsible for the formation of ATP. It plays a big role. This is what we saw on the mitochondria inner membrane, where we saw those stalk particles. These are the stalk particles that we spoke about. So this is the electron transport chain. As we can see, if we look at these two diagrams, we can see this is occurring in the inner membrane, the cristae, right? This inner membrane is where these reactions are taking place. The outer membrane of the mitochondria is out here, right? And so there's a space between the inner membrane and the outer membrane. You see this reddish area here. This is the intermembranal space. And what you want to have happening is hydrogen ions will move from the matrix, and go across that inner membrane into the intermembranal space. This is facilitated by these molecules, these protein molecules that are embedded in the membrane, in the inner membrane. And the passage of the electron along this membrane is facilitating the movement of the hydrogen ions across. Why is it important? What we want to have happen here is an electrochemical gradient has to build up, where hydrogen ions from inside of the matrix will move across to the intermembranal space, and so increase the concentration of hydrogen ions there. And that in itself would generate ATP through ATPase. Good. We have here NADH, which is the reduced form of NAD. Remember that would have picked up the electrons and the hydrogens from the glycolysis, from the link reaction, from the Krebs cycle. And now they have brought it to this electron transport chain, and the electron is going to pass across. This molecule will pick up the electrons and the hydrogen ions, and they will become reduced. It will become reduced, and the molecule will become oxidized. The good thing is this now can go back to glycolysis. It can go back to the link reaction. It can go back to the Krebs cycle and do the same thing, pick up hydrogen from the molecules that are there. So this keeps the process going. You only have a limited amount of NAD plus, NAD ions, and so they have to be recycled continuously. So anything that disrupts this process, any medicine, anything that you take in your body that will disrupt this process, eventually will shut down your cells. Your cells will not be able to get enough energy to do that. And cyanide is one of those things that can do that, right? And this is where your knowledge of biology will help you in understanding how medicine works and how we are able to solve the problems that we are faced with, right? Application of knowledge. So as the electron pass across, one of the molecules will pick up the electron, pick up the hydrogen, and become reduced. And this first one now will become oxidized. And therefore, this now moves it around, because some of these proteins, they are fixed in the membranes, and some are moving like these here. You are not required to know the names of these proteins at this level. At university level, you do, right? But you just have to have a general appreciation of the molecules that are involved. You have four main ones that are involved in the movement of the electrons and hydrogen ions and the eventual reduction of oxygen to form water. That is why it's so important that oxygen must be present. If there is no oxygen, there is nothing to be the final electron acceptor. Oxygen is the final electron acceptor to form water. So if we do not have oxygen, then this whole process would not take place. From glycolysis onwards will not take place. They will only have up to glycolysis taking place, right? So oxygen is very important. Now, as the hydrogen ions move across and they build up here, the only pathway they will be able to move through to come back in is through this molecule here called ATP synthase or ATPase. It's a protein that facilitates the transfer of the hydrogen ions down an electrochemical gradient. This doesn't require energy. This is not an active process, right? And you have the formation of ATP. So you are going from a molecule that is rich with energy to form smaller packets of energy. So for every one molecule of glucose, you produce 38 molecules of ATP. Again, this is just showing you it in a different way. So let's do the maths. Let's look at the net products of aerobic respiration. We know that there are many different pathways, many different areas that we had ATP being produced. We had, of course, glycolysis. You had we had the link reaction. We had the Krebs cycle. From glycolysis, we have 2 ATP being produced directly. Remember, we put in 2, but we got out 4, so therefore we had 2 with the net production directly. Then we had 2 NADH. We had 1 for each molecule, so since we had 2 and each one is giving us 3 ATP eventually in the electron transport chain, we therefore have 6 ATP. So both of them would come from glycolysis, but we only get in this one directly. This one we will get eventually in the electron transport chain. So that will give us 8 ATP. 8 ATP coming from glycolysis. What about the link reaction? We have, from the link reaction, remember we would have lost carbon dioxide and we would have had NAD being reduced. And since we have 2 molecules, you have 2 NAD, and therefore you have 6 ATP here because we have 3 for every NAD. So this will give us 6 ATP. And in the Krebs cycle, remember we had 1 per molecule being directly produced, but we had 2 molecules, so there you get in 2 ATP being produced directly. And then we had 3 NAD, but we had 2 molecules, so literally 6 NADH. So you have 6 NADH, and that would convert 6 trees are 18 ATP. That's coming from the NADH. And what about the FAD? We had 2, and since each one is going to give us 2, so you're literally going to have 4 ATP from the NAD. Sorry, from the FADH. So here we have 18 and 2, which would be 20. And here we have 4. So when we add it up, we have 20, 26, 30 and 8, and so this is where we get 38 moles of ATP. Good. So you need to have a working knowledge of this. You need to be able to, let me just put it a little clearer. I don't have to write ATP again. We know it's all ATP. Right? So your maths here, you have 38 moles of ATP. So this is for every one glucose molecule. You are getting 38 moles of ATP. So this was why this is why it happens. This is why oxygen is so useful to carry out aerobic respiration, because we're going to yield more energy and our body is going to get more energy available to do work. When we have anaerobic respiration, our body is only going to produce 2 moles of ATP for every glucose molecule we would have had. Remember, know what is the direct production, these two, through substrate level phosphorylation. The others will come in through oxidative phosphorylation. These here will come in from FAD, where we only get 2 for every FAD, and the others will come in from NAD, where we get 3. There are different schools of thought as to exactly how much, whether it's 2, whether it's 3, or slightly less, but CXC will accept these numbers, and you can use that 38 moles of ATP for every one glucose molecule. So here we have a tool, a simple respirometer, which the syllabus says you should know. There are some important parts there that you must appreciate how they are used in carrying out this experiment. What we are trying to do here is to measure the uptake of oxygen. So we know living organisms, as we recognize, will carry out aerobic respiration, and so they will be taking up oxygen. But we know they will also be producing carbon dioxide. So if we keep both of them in there, we would not be able to measure and see the way in which the oxygen is moving. But if we remove the carbon dioxide by this structure here, which is potassium hydroxide, which is otherwise known as soda lime, if we have this there, then should this organism be respiring and producing carbon dioxide, this liquid would absorb it. So you can either have the liquid or you can have some pellets. Sometimes you may see it above. Of course, that substance is very toxic, and so it must not come into contact with the living organisms. You know, at all times you try to preserve the health of the organisms. You also should not be touching this with your hands. You should wear gloves and so on. So if they ask you to set up an experiment, you will have to appreciate the things that you will do to try to get the best possible results. You must not allow the pellets to touch the sides of the tube, so you can use something where you can direct it down into the tube. You also have at this stage, this area here, a syringe. This literally is acting as a reservoir to facilitate the calibration of this part of the apparatus. This is the capillary tube, and this will have air within it. So what you want to do is to pull up a colored liquid within, so at the end of this tube, you will literally have this end dipped into a colored liquid. And by maneuvering the syringe, you can pull up the colored liquid at the end of the capillary tube. As you are absorbing carbon dioxide within the soda lime, whatever carbon dioxide is being produced on respiration, then the oxygen, the air that is in here that has oxygen, is going to be moving in. The organism is going to be taking in oxygen. And so you can chart the movement of the bubble as it moves along the capillary tube. And since you know the diameter of the capillary tube, you can actually work out the area, and you can work out the volume of oxygen that is taken up over time. So you can take readings, you know, at a minute or 5 minutes or so, and you can measure the rate of oxygen uptake. This is a living organism. You can do the same with something that is non-living and compare them So this is a simple respirometer. You also have another type of respirometer here. This is a differential respirometer. And here you are seeing they're connected by a U-tube manometer, and you have the manometer fluid. So you have the manometer fluid within here, and this fluid will allow you to see the movement of the gas, right? So you would start off, when you start off, both sides will be at the same point. Some features of this apparatus, you will notice that the whole apparatus is within a water bath. This helps to regulate and stabilize temperature and pressure, so that those things are not influencing the rate of the oxygen uptake. What will happen in this apparatus, we have two sets of things. We have the glass beads within this tube, and so this tube is acting as the control, right? It's acting as a control. And this one, where we have the respiring organisms, they will be taking up oxygen and giving off carbon dioxide. The soda lime will absorb the carbon dioxide, and so once they are respiring, they have to get their oxygen from somewhere. This is closed off at this point, so that the only place they can get oxygen would be from within this tube. And so this liquid will move in the direction where respiration is taking place. So again, you must know be able to draw and label or identify the different features of this entire apparatus, and be able to explain how it can be used to show and to be able to carry out an experiment to measure the uptake of oxygen by living organisms. So we have been going on and on and on, and certain at this point you are well able to answer some CAPE type questions. So let's look at a few CAPE type questions. Now I know people may say, well, why don't you have more multiple choice questions? We have had multiple choice questions in the past, and by all means, do multiple choice questions. But at the end of the day, whether it's multiple choice, whether it's structured, whether it's essay, you need to be able to get a good grasp of the concepts. You need to be able to answer any format a question can come in. So I urge you, please, yes, do multiple choice questions, but don't just do it to cram off an answer. Do it with a fair understanding of, how to apply your knowledge. And so, yes, in doing these questions, it will still help you. Still, do not, put off doing these questions, because if you're in lower 6, then you're going to be doing it next year again. If you're in upper 6, you're going to be doing it at university level. So do not totally abandon doing structured papers. They actually help you to get a better grasp, and so you can apply it when you do the multiple choice type questions. So here it's asking you, they're showing you a part of the mitochondria, and they're asking you to identify the structures labeled A, B, C, and D. Good? So if we see A, what do you think A is? Look around clearly. You see an A, and it's in the inner part, and so this will be the matrix. This will be the matrix of the mitochondria, that innermost liquid that was within the inner membrane of the mitochondria. And then B, right? These will be the cristae, the enfoldings of the inner membrane. So this is the cristae. And then you have C, which you would see these are really ATPase, and we can put the stalked particles. I apologize for my writing here. This challenges me every time. Yeah, it's not quite as easy as it looks, right? D, as you will know, the mitochondria, remember it was once a living organism, free living, so it has its circular DNA. So you will put there circular DNA. So these would have been identified. Use an arrow labeled X indicates the site of the Krebs cycle. Using an arrow labeled X indicates the site of the Krebs cycle on figure 1. As we know, the Krebs cycle takes place in the matrix. So you have this area here. Oops. Right, so we can have it here. Using an arrow, please, whatever they tell you to do, do it. If they say an arrow, do not just put a line. And we say to labeled X, so you'll put X there, right? And so that would have answered that question. Here, it is a flow chart which represents part of glycolysis pathway. And you would see it's that first part where energy is being used up, right? The investment part of glycolysis. And they had the different numbers, 1, 2, 3, 4. And firstly, the first question, in which part of the cell does the process of glycolysis take place? Easy pickings, right? Of course, this is the cytoplasm. The cytoplasm. So you should have written there cytoplasm. The next part of this question is on the next page. So when I remove it, you would not see this. But as we look here, they literally ask us to explain what is happening here at the different parts. So I think each question is worth 2 marks. No, it's actually worth half of a mark. So it's worth half a mark. So you state what is occurring at each of the stages labeled 1, 2, 3, and 4 in figure 1. So you have to state what is occurring. Simply what is occurring, not why it's occurring, what is occurring. And so here, we are going from glucose to glucose 6-phosphate. Phosphate is being added to the molecule. So ATP is being broken down to ADP, and the phosphate is going in here. You have the enzyme that is responsible is called a kinase. So we are saying they asked us what is happening here. So we said ATP is being used up, and a kinase is responsible, the enzyme that is responsible. Then you go from glucose 6-phosphate to fructose 6-phosphate. What you have here is an isomerase. So you have isomerism involved, where you are converting the molecule from the structure of the molecule from a aldehyde to a ketone. Then from here to here, what is taking place is that you are adding, so there's another kinase involved here. So just like you have up here, you're going to insert the same answer at this point. A kinase is involved, and you are converting, the molecule is being phosphorylated. And then at 4, what you are having is the molecule being split from a 6-carbon molecule to 2 3-carbon molecule. State what is, this is what we just did. Suggest the significance of the pathway shown in figure 1. Why is this molecule, why is this pathway so important? As we indicated when we were doing the actual pathway, it is important for glucose to be fixed in the cell. If the phosphorylation did not take place, the glucose could have easily left the cell. As the phosphate molecule is added to glucose, it enlarges the molecule, and it makes it more difficult to get out. Also, it allows the molecule to become more reactive, and so the process will take place faster. You also had this process occurring to produce this 3-carbon molecule, because we needed to form pyruvate for us to be able to go into the link reaction within the mitochondria. So that pathway was very important to break down that glucose molecule and to yield the products that we yielded. Again here, the question is asking for you to be able to label the stages, all right, where the following would be happening. C to indicate the decarboxylation reactions. So where is carbon coming off? We know a carbon will be coming off here, right? So you have a C label there. You will have a carbon coming off here from the 6 carbon to the 5 carbon. So you have a C there. You will have a carbon coming off here. So you will have another C there. And H to indicate dehydrogenation reaction. Dehydrogenation, simply the loss of hydrogen. You will have hydrogen, let me use a different color. You will have hydrogen coming off here. Of course, you remember NAD is being reduced. So you will have NAD. So they just tell us to put an H. So I'm just going to put an H. So we have here H to indicate that a hydrogen is coming off. Of course, we will have hydrogen coming off here. You have hydrogen. Again, you're forming NADH. You also have H coming off here with the FADH. And you have another one coming off here. Right? Remember, you have 4 hydrogen within the Krebs cycle, and so 4 NADH and 1 FADH. And you had 1 NADH up in the link reactions. So this should have been your diagram. Say exactly where the Krebs cycle occurs in a cell. Well, we did see it in the other one, but this is a different paper. Again, the matrix of the mitochondria. That is where the Krebs cycle occurs, the matrix of the mitochondria. Briefly explain the role of NAD in the Krebs cycle in relation to energy production. So briefly explain the role of NAD in the Krebs cycle in relation to energy production. Remember, NAD is going to accept an electron and accept hydrogen and become reduced to NADH. That now will allow it to go into the electron transport chain, where the electron and the hydrogen would pass along and will result in the formation of ATP. So NAD is very important in moving hydrogen, moving energy towards the production of energy, the formation of ATP within the electron transport chain. Define the term phosphorylation and distinguish between photophosphorylation and oxidative phosphorylation. Okay, so I brought this in even though we are not doing photophosphorylation at this point, because that really occurred in photosynthesis. It's important, as I said, you can be asked these questions. It's important for you to be able to identify the different types of phosphorylation. So phosphorylation, as we said, is simply the addition of phosphate to a molecule. But photophosphorylation is when you have water becoming oxidized to hydrogen ions and oxygen, and the energy that is captured within photosynthesis is now being used to form ATP. That is photophosphorylation. Whereas in oxidative phosphorylation, you will have the oxygen being reduced. So in one, the oxygen is being, the water is being oxidized, and the other one, oxygen is being reduced, and the formation of ATP, which is being driven by redox reaction, by reduction followed by oxidation, transferring energy along, and then finally being accepted by the electron acceptor, which is oxygen, to form water. So in one, you have the energy being driven from photosynthesis, from the sun. The other one, the energy is being driven from what was captured in the glucose molecule to eventually the electron acceptors, NAD and FAD, and NADH and FADH, to finally be used in the formation of ATP, oxidative phosphorylation. So you are joining a P to ADP to form ATP, but it's how that is being done, where it is coming from, where the energy is coming from to cause this process to take place. Discuss two similarities and two differences between the chemical reactions which occur during photophosphorylation and oxidative phosphorylation. Well, I just mentioned them. You know, in photophosphorylation, water is being oxidized to yield the hydrogen and the oxygen. In the other one, in oxidative phosphorylation, the oxygen is being reduced to form water. You also have the sources of the energy, where the energy is coming from. In photophosphorylation, it's coming from the sun, all right? And in oxidative phosphorylation, it is coming from these molecules which would have captured the energy initially coming in glucose. Two similarities, both of them are producing ATP. Both of them are producing ATP. And another similarity is both of them involve electron acceptors. Of course, in photophosphorylation, you have NADPH involved in moving the energy along, whereas in oxidative phosphorylation, you have NADH, not NADPH. NADPH would be in photosynthesis. Good. So there are more. We can look at others when we talk about, you know, the enzymes that are involved and so on. So we would have looked at the different objectives we set out to achieve at the beginning of this course. Recall the structures, the locations, so the structures in mitochondria, the locations, whether it's in the cytoplasm or in the matrix, and so on, the metabolic reactions, glycolysis, the Krebs cycle, the link reaction, the electron transport chain involved in the process of aerobic respiration. Review the stages of glycolysis, the Krebs cycle, and I would add the link reaction and the electron transport chain. So go over them, look, make sure you understand, make sure you can explain what is happening, make sure you can justify the significance and the importance of these processes in ensuring energy is being released within this process. Draw, label, and explain respirometers. So we would have looked at two different types of respirometers. Go back, make sure you can draw it and label, and how you can explain these experiments that you should have done in school. These are required experiments that you should have a fair working knowledge of. Appreciate the importance of aerobic respiration to all living organisms. So, you know, we literally depend on energy. We all, you know, we cannot capture that energy ourselves. We have to depend on autotrophs to capture that energy. In turn, we either will feed on the autotrophs or we feed on a heterotroph that fed on the autotroph. And so eventually we need energy. Every metabolic process in our body needs energy. Once the cell is alive, it needs energy. And so even if somebody is brain dead, they still have to be on a respirator because their body still needs oxygen for all of these systems to work and so on. So living organisms need energy, and the better way for them to get that energy is from the process of aerobic respiration. In the next session we will be looking at anaerobic respiration. What happens when there's no oxygen present? What happens in a plant? What happens in an animal? What happens in microorganisms? So please, Please interact with the work. It's not difficult. You need to sit, you need to see its application in its many different forms, right? And I'm sure your teachers would be very, very willing to assist you, to answer questions and so on. The Division of Education is trying to put things in place for you to be able to facilitate all these videos on their website. Please, they are already on the website, so go to the Division of Education's website and get the videos, go over them and make sure you understand them. I am so thankful that I'm given this opportunity to be able to speak to you and to try to, give off whatever little that I know. Keep studying and all the best in your exams going forward. From the Division of Education, Innovation and Energy, I am Ms. Jamilla. I'm in Bacchus. Thank you very much for viewing.